Good day everyone and welcome to Microbiology and Parasitology this summer. I am Dr. Sipache Basit, the Dean of the School of Medical Technology, and I'm very glad that you are attending these online classes here in Microbiology and Parasitology, and I hope that you will learn a lot this particular sem. So to start, I will be discussing Introduction to Microbiology. So our official textbook is actually Burton's Microbiology for the Health Sciences. So, Introduction to Microbiology, we'll talk about what is microbiology, why do we need to study microbiology. We'll also tackle about the first microorganisms on Earth, the earliest known infectious diseases, the pioneers or important scientists in the science of microbiology, and what are the possible careers in microbiology. So, first, what is microbiology? Microbiology is a branch of biology, and as you all know, biology is the study of living microorganisms. So, what we're studying here in microbiology is actually microorganisms. So, it is like an advanced biology course wherein we study microbes. So, when we say microbes, these are extremely small organisms, so small that they are said to be microscopic, which means that you cannot see them using your naked eye. So yet they are living organisms, and there are also certain microorganisms that are considered as non-living entities, meaning to say they are not living things. Examples of these are the viruses and prions. Of course, as we all know nowadays, one of the most popular viruses is COVID-19. So this is the disease is COVID-19, and it is caused by SARS-CoV-2. So it's a coronavirus. Okay, so it's an example of a virus, and virus is considered to be as a non-living entity. So living microbes are known as cellular microbes, which means that they are made up of unicellular, meaning they are single-celled organisms. Examples are bacteria, archaea bacteria, and then algae, protozoans, and even some fungi. So, non-living microbes are acellular microbes or infectious particles, which may include viroids, prions, and viruses. So, microbes are ubiquitous. So, when I say ubiquitous, they are found virtually everywhere, even the skin, in your clothes, the surface of your table. So microorganisms are present. So if we're going to look at the organizations of this particular chart, microbes are divided into two major categories. So we have the acellular infectious agents. So prions or the prions are actually one of them. So prions are also known as the infectious proteinaceous molecules, because they are actually made up of protein molecules. If you are familiar with the mad cow disease, so mad cow disease is actually caused by prions. Okay? Viruses, well, one of the best way to describe viruses is that they are considered to be as obligate intracellular parasite, which means that they cannot live outside our body. Okay? They need a living host in order for them to survive, in order for them to replicate. So, which means that coronavirus is an example of a virus that needs to be inside the human body in order for them to replicate. Okay? They cannot stay in the environment for so long, so they will not be able to replicate. So that's the reason why one way to prevent the transmission of COVID-19 is to use proper PPE, so that these viruses will not be able to transfer from one host to another. That way, we'll be able to somehow minimize local transmission. And then talking about cellular microorganisms, there are two types of microorganisms. So we have the prokaryotes and the eukaryotes. So when you say prokaryotes, pro means primitive, karyo refers to nucleus. So when you say prokaryotes means these are organisms that have primitive nucleus. So when you say primitive nucleus, it means that they don't have actually nucleus because they do not have nuclear membrane. So example of these are the bacteria and the archaeobacteria. What's the difference? Bacteria has cell wall. Okay? So that cell wall is made up of peptidoglycan. Okay? Archaeobacteria does not have cell wall. So archaeobacteria lives in extreme environment. So some of them are methanogens, meaning to say methanogens are capable of producing methane. And then we also have the extremely thermophilic. So that's the second type of archaeobacteria. Extremely thermophilic lives in environment that is high in temperature, extremely high in temperature, such as the benthic surface of the ocean, geyser, volcano geyser, hot springs. And then we also have extremely halophilic. So when you say extremely halophilic, it means that these particular organisms would require high salt concentration. So again, there are three types of archaeobacteria: methanogens, extremely thermophilic, and extremely halophilic. And then we have eukaryotes. The prefix "eu" refers to true, which means eukaryotes means true nucleus. And the reason why we call them true nucleus is because they have a real nuclear membrane. So humans, such as us, although we're not microorganisms, okay, we are eukaryotic because we have a true nucleus. So speaking of microorganisms, eukaryotes are the algae, fungi, and protozoans. Okay, so moving on, have you ever wondered when you were a child and then your mom would tell you that, oh, don't touch that filthy thing, it is covered with germs. So sometimes when we drop food on the floor, and then you will actually pick it up and you'll say, wala pang 5 seconds. The 5 seconds, so 5 second rule. Well, it doesn't really apply at all. But because what I told you a while ago, microorganisms are ubiquitous, means that they are everywhere. So the microbes that we should be very cautious with are called germs. Okay? Microbes that can cause diseases are sometimes referred to germs. So the scientific term for disease-causing microbes is pathogens. So we call them pathogens. So when you say pathogens, these are microorganisms that are capable of causing diseases. So microbes that do not cause diseases are called non-pathogens. So they are not harmful. In fact, majority of them are beneficial. Let's say, for example, probiotics, Lactobacillus shirota strain, the one that is found in probiotic drinks such as Yakult. So they are not pathogenic. In fact, they are helpful in maintaining normal microbiota of gastrointestinal tract. The good thing about it is that the vast majority of microbes that we have in the world are not pathogenic. They are non-pathogens. However, in this particular course, we will be dealing with pathogenic microorganisms because we shall be discussing microorganisms that are medically important. Okay, so microbes that live, okay, when you say on, such as those that are found in skin or inside our bodies, we call them indigenous microbiota. Before, they call it normal flora. But since microorganisms are not plants, so we avoid calling them normal flora. So we call them indigenous microbiota or sometimes normal microbiota. So some members of our indigenous microbiota are opportunistic pathogens. So they have the possibility of becoming opportunistic. What does it mean? It means that they can take advantage of hosts with weak immune system. So who are examples of people with weak immune system? Those people who are diagnosed with HIV and eventually they have AIDS. So they have weaker immune system. Who else? People who are undergoing chemotherapy, because chemotherapeutic agent will actually kill the cells of your immune system in the process. People who just receive newly transplanted kidneys because they are drinking immunosuppressive drugs so that their immune system will not reject the newly transplanted kidneys. So, as a result, they become immunocompromised. Geriatric patients, pediatric patients. So, their immune system may not be functioning as compared to you guys. So, they are considered to be as people who may be at risk of getting opportunistic infections. Okay? So, opportunistic pathogens are microbes that can cause disease, but usually they do not. For people who have normal immune system like you and me, but they can be thought as microbes that are awaiting the opportunity to cause the disease. Meaning, they're just waiting for the right moment for them to inflict disease. Therefore, people who are immunocompromised should be careful. They should be watchful in things that they're doing. So, for example, patients who have just received a kidney transplant, and then they are drinking immunosuppressive drugs should be wearing masks most of the time. Okay? So, pathogens, therefore, can be categorized into two types. So, those that can cause infectious diseases, and sometimes, some of them may even cause microbial intoxications. So, what's the difference? So, when the organisms can cause infectious diseases, so what will usually happen is that the pathogen will colonize your body. And after the pathogen have colonized your body, it will cause disease. And then, this type of disease is known as infectious disease. Example, MRSA infection. You know what MRSA is? MRSA stands for methicillin-resistant Staphylococcus aureus. So this particular type of organisms is drug resistant, which means that Staphylococcus aureus is usually easy to manage, but if they become MRSA, you need a certain drug in order to combat them, because they have already established resistance, for example, to penicillin. And then gas gangrene. Gas gangrene is an example of the infectious disease caused by Clostridium perfringens. By the way, guys, I will be talking about scientific names in my lecture. So if I were you, as you follow my lecture, you may pause the video and then look for the proper spelling of the organisms that I'm saying. Okay? So for example, gas gangrene is caused by Clostridium perfringens. Okay? Clostridium perfringens. So that is an example of infectious disease. So which means that there is colonization. So in order to start infection, there should be colonization. On the other hand, when we say microbial intoxication, a pathogen produces a toxin in vitro. Okay? So what does it mean by in vitro? When you say in vitro, it is outside the body. So which means that the pathogens, even before we ingest them, have already produced toxins. For example, a pasta that has been prepared unhygienically may have Staphylococcus aureus. So, as the pasta is actually prepared unhygienically and has been sitting on the table for quite some time with an ambient temperature, what will happen is that Staphylococcus aureus will multiply. As they multiply, they produce toxin. So, during handaan, a person will ingest the toxin. So, the toxin will cause a disease. So, the type of disease is called microbial intoxication. So, staphylococcal food poisoning is one of the most common examples of food poisoning. But so what you'll feel, there would be usually nausea, vomiting, abdominal cramps. But the one that is more deadly is the one caused by Clostridium botulinum. So it causes foodborne botulism. Do you know that the botulinum toxin is one of the most potent toxins in the world? It is even more potent than the cobra venom. Okay, so Having said that, now I hope you'll be able to realize that studying microbiology is important. It's because microbes play significant roles in our lives. Without microbes, I find it, I'll be able to deduce that we'll not be able to survive this planet because they are essential for life on this planet. Particularly, there are some microorganisms that are said to be photosynthetic. Photosynthetic algae, photosynthetic bacteria such as cyanobacteria, they're essential. They are producer in food chain. They produce much oxygen in our atmosphere. Microorganisms are also involved in the decomposition of dead organisms and waste products, particularly fungi. Without microorganisms, you'll be seeing dead bodies lying around. Saprophytes are organisms that live on dead and or decaying organic matter. Mushroom is an example of fungi, and it is an example of saprophyte. So the use of microbes to clean up toxic waste, particularly sometimes you may even hear of oil spills in the ocean, okay, and other industrial waste. So some microorganisms may clean this oil spill by using their metabolic prowess, okay. So we call it bioremediation. So that is a very important process, okay. So here's what I'm saying. So saprophytes, such as mushroom, okay, and a decaying wood, it's an organic matter. Saprophytes will help the environment by decomposing these organic materials. So in doing so, byproducts such as nitrate, phosphates, and sulfates, ammonia, CO2, water, and other chemicals will be embedded in the soil, and this makes the soil much richer. And because of this, there are some nitrogen-fixing bacteria in the soil, and this will allow plant to propagate much better. So that's the circle of life. Okay? So another reason why do we need to study microorganisms? So as what I've shown you a while ago, many microbes would play essential roles in various chemical cycles such as the carbon cycle, nitrogen cycle, oxygen, sulfur, and even phosphorus cycles. There are some microorganisms such as algae and bacteria, and they serve as the producer. So in food chain, so they are usually the first one to be eaten up. Of course, since they're small, they will be eaten up by tiny animals. So they are important links in food chains. Microbes that live in intestinal tracts of animals aid in the digestion of food and produce beneficial substances. So that's the reason why cows, they are able to metabolize the grass that they are eating because of the presence of ruminants. These are bacteria found in the intestinal tracts of cattle. Okay? For many years, microorganisms have been used as cell model. So the more the scientists learn about the microbial cells, they actually learning more about cells in general because they're using these microorganisms as models. So here is another example of nitrogen cycle by nitrogen fixing bacteria. So what will happen here is that nitrogen gas is found in our atmosphere. So they shall be absorbed, okay, by the plants. And here in the soil, we have nitrogen fixing bacteria. And with the presence of ammonia, nitrate is produced, and this will replenish the soil nutrients. So nitrogen returns back to air. However, because of the presence of the nitrates in the soil, which enriches the soil, serves as the nutrients for plants, so many of the plants will propagate. And one of the manifestations that you can actually see that there are indeed nitrogen fixing bacteria in the soil is that when you pull out or pull up these legumes, you'll be able to see nodules in the roots. So the presence of nodules in the root is one of the manifestations that they are indeed nitrogen fixing bacteria in the soil. Okay? So another reason why we need to study microorganisms, they are actually useful and helpful in industries, in food, beverage, chemical, antibiotic industries, and also in genetic engineering. So genetic engineering is another important field of microbiology because here we'll be able to utilize gene from one organism and then we insert that gene into another cell. So we call that as the vector. And vector could be a bacterial cell such as Escherichia coli or yeast cell. And the cell that receives the new gene is then capable of producing new gene products coded for by the new gene. So because of this, we'll now be able to produce insulin, for example. So the use of living organisms or their derivatives to make or modify useful product or processes, we call it biotechnology. Okay? So I'm just listing some of the important roles of microorganisms. There are millions of roles of microorganisms. There are millions here in the universe, and as what you can see here in the illustration, that is an example of a food chain. So going back to history, what are the first microorganisms on Earth? Well, many scientists have already dated it back about 3.5 billion years ago. So the fossils of primitive organisms date back about 3.5 billion years. So they cannot really specify which organisms were the first ones, but candidates for the first microorganisms on Earth are Archaeobacteria. Remember what I told you about Archaeobacteria? And Cyanobacteria. Cyanobacteria are the blue-green algae. So, infectious diseases of humans and animals have existed for as long as humans and animals have inhabited the planet. However, the earliest known account of pestilence occurred in Egypt about 3180 BC. There is one important theory that during the olden days, many scientists have debated about: spontaneous generation theory. You know how silly spontaneous generation theory is, but anyway, I'll explain it to you. During the olden days, many people believed that microorganisms can actually exist from non-living cells. Literally, if they saw a frog emerging from the mud, they would say that that mud gives rise to frog. Something like that. Okay? And one of the most important or one of the most famous proponents of spontaneous generation theory is John Needham. So John Needham experimented on chicken broth. So he set up two tubes. The other tube, he allowed it to be exposed without placing any cover on top of it, and then the other one, he covered it and then exposed the chicken broth. And then, lo and behold, after several days, he noticed turbidity in both tubes. Turbidity is actually an indicator that organisms have grown in the tubes. Okay, so what's wrong with the experiment of John Needham? Okay, well, one of the flaws the major flaw in John Needham's experiment is that he failed to sterilize the equipment. So, because of the fact that he failed to sterilize the equipment, okay, so the chicken broth in the first place, even before incubation period, had already been contaminated. Which means that the lesson here is that whenever we do experiment, we always use sterile equipment because we do not want any form of contamination. Okay? So, during these heated arguments on spontaneous generation theory, there was this one particular... Then we have a guest. Anyway, one of the most important proponents of people who are against spontaneous generation theory is Francesco Redi. So Francesco Redi experimented and he was trying to prove that maggots did not come from a piece of meat. So he initially set up two 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 plates. Okay, so one is exposed, and the other one is actually covered. So there are two setups. Okay, so he placed piece of meat, and eventually, of course, this meat decayed, and maggots came out from this exposed plate, because flies would actually touch the decaying meat, and then the other one is actually covered. So obviously flies will not be able to touch the decaying piece of meat. So what will happen here is that of course there won't be any maggots, right, baby? So since there are no maggots in this particular meat, so at least somehow Francesco Redi was able to prove that maggots did not come from this decaying piece of meat. However, during this time, Antoine Lavoisier, I hope I was able to pronounce his name correctly, discovered the importance of oxygen in human life. And those who believe in spontaneous generation theory argued that since you covered the jar, it wasn't, they were not plates pala, jar pala. Since you covered the jar with a cover, sorry, and then you did not allow oxygen to come into contact with a piece of meat, and oxygen is a vital element of life. And then Francesco Redi modified his experiment. So instead of using a totally covered jar, Francesco Redi utilizes a small piece of net, and this small piece of net will allow air to come in, but it will not allow the fly to come into contact with the decaying piece of meat. So lo and behold, the result of the experiment, no maggots came out from the decaying piece of meat. But the arguments between those who believe the spontaneous generation theory did not stop until there was this one man. So it was Louis Pasteur. Okay, so what Louis Pasteur did was he utilized a flask, and then he filled the flask with chicken broth, like the one that was utilized by John Needham, and then he bent the tube of the flask in such a way that it becomes like a long S. So he called it the swan neck flask, because it appears like the neck of a swan. So this will prevent microorganisms in entering. This prevents the microorganisms in the air from entering the flask, yet at the same time it allows air to flow freely. So what you would expect, by the way, is that microorganisms will be trapped here at the convoluted portion of the lung of the swan neck flask. So if the swan neck was broken, microbes could enter the flask and grow. But those unbroken flasks, in unbroken flasks, microorganisms were not able to contaminate the broth. Okay? Because of this experiment of Louis Pasteur, so Louis Pasteur was able to disprove the spontaneous generation theory. And because of that, Louis Pasteur became the father of microbiology. So there are many contributions of Louis Pasteur in the science of microbiology. So he was a French chemist who made numerous contributions in the field of microbiology. So Louis Pasteur investigated in different fermentation products. Fermentation is the process of anaerobic breakdown. So when we say anaerobic breakdown, in the breakdown of let's say for example, carbohydrates in the absence of oxygen. And one of the byproducts of anaerobic breakdown could either be acid or alcohol, depending. If it's yeast, then one of the products would of course be alcohol. So because of the science of fermentation invented by Louis Pasteur, French becomes one of the best winemakers in the world. So there are many products that are actually the results of fermentation. Cheese is one of them, beer is one of them. Okay, and then Louis Pasteur was also able to develop the pasteurization process. Pasteurization is very important because before they are drinking milk even without undergoing pasteurization, and there are several milk-borne pathogens. Okay, so and pasteurization is actually application of enough heat to kill the pathogens in the milk. And Louis Pasteur discovered life forms that could exist without oxygen. We call them the anaerobes. And he was able to develop several vaccines, including the rabies and the anthrax vaccines. Okay, so wow. For a man who lived between 1822 to 1895, these 5 bullets are actually accomplishments that were not achieved by any other man who lived with the same lifespan as Louis Pasteur. And then another contemporary of Louis Pasteur, although much younger, he was born in 1843, Louis Pasteur in 1822, Robert Koch. So, a German physician who made numerous contributions to microbiology as well. Of course, one of the most important of which is the germ theory of disease. We call it the Koch's postulate, and he discovered the Bacillus anthracis, the bacteria that can cause anthrax. Okay, he discovered that it is able to produce spores. So he also developed methods for fixing and staining bacteria, and he was able also to help by developing methods to cultivate or to culture bacteria. So here's Koch's postulate. So exciting. But before that, I'd like to share my DIY slide, not this one, on how Robert Koch was able to discover the Koch's postulate. Okay, so let's read about Koch's postulate first. Okay, so it says here, if an organism fulfills Koch's postulate, it has been proven to be the cause of that particular infectious disease. So Koch's postulate proved the germ theory of disease. And Robert Koch gave a tremendous boost to the development of microbiology by stressing laboratory culture and identification of microorganisms. Okay, however, there are some circumstances that do not fulfill the Koch's postulate. So an example is the COVID-19 disease that we're having right now. It doesn't follow Koch's postulate because coronavirus is not cultivable. We cannot culture coronavirus. Anyway, let me tell you the story how Robert Koch was able to discover his own postulate. So while walking in the field, Robert Koch discovered a dead cattle. It's dead. Why did I say it's dead? Makabaligtad. Wala namang buhay na cattle na ganyan, di ba? Tsaka may X. Okay? So what Robert Koch did, he extracted the blood of a dead cattle, and since he was able to find a way to culture bacteria, so he cultivated the bacteria. And then the pure isolate that he got from the blood of the dead cattle, he cultivated it, he injected it into another animal. And the same animal died from anthrax. And he was able to re-isolate the bacteria from the blood of this particular dead animal who died from the same disease. Okay? So here's Koch's postulate number 1. The microorganisms must always be found in similar diseased animals, but not in healthy ones. Number 2. The microorganisms must be isolated from a diseased animal and grown into pure culture. Number 3. The isolated microorganisms must cause the original disease when inoculated into a susceptible animal. And number 4, the microorganisms can be re-isolated from the experimentally infected animal. So, which means that Koch's postulate is so important because here, he was able to prove that for every disease, there is a causative agent or an etiologic agent. And that causative or etiologic agent can be transmitted into another healthy subject if you will be, for example, injecting or infecting the healthy subject with the same causative or etiologic agent by allowing it to be cultured first. However, in modern days, they were able to discover that not all disease infectious process or disease infection process will follow the same Koch's postulate because there are some microorganisms that are not cultivable. So an example of this is the SARS-CoV-2, which is the causative agent of COVID-19. We cannot cultivate them, but the only reason why they were able to discover that it is indeed coronavirus is through molecular diagnosis and of course by electron microscope. Okay, so there are many careers in microbiology. So most of you who are listening to this screencast or watching this screencast, you are in health sciences. So could be a medical technologist, could be a nurse, pharmacist, physical therapist. So yes, microbiology is important in your discipline. But there are some people who may be considered as a hardcore microbiologist. They are scientists who studies microbes. So there are many career fields in the science of microbiology. You can actually be studying bacteria, bacteriology, phycology is the study of algae, protozoology is the study of protozoans, mycology is the study of fungi, parasitology is the study of parasites, and then virology. Okay. However, there are many fields in microbiology. It could be industrial, environmental, food, aquatic, soil. But here in this particular course, we will be dealing with medical microbiology. So it involves the study of pathogens, the disease they cause, and the body's defenses against disease. So it is concerned with epidemiology, the science or studying the pattern and spread of the disease, transmission of pathogens, disease prevention measures, aseptic technique. Aseptic technique is very important because here you are managing risk. Okay, treatment of infectious diseases, immunology, and of course, production of vaccines. Okay, so that ends the introduction to microbiology. I hope you will enjoy microbiology in as much as I enjoy it too. So again, please do not forget to buy books. I've actually pasted the link of the book in the announcement, okay, of the Bright Space. So with that, I'd like to greet everyone. Good day. Stay safe in your respective. God bless everyone.